Hello, this is Ashman. You're watching Ashman videos, and today we're talking about the cell cycle control systems and internal and external factors. The cell cycle is a complex set of stages that is highly regulated by checkpoints in the cell cycle control system. The cell cycle consists of interphase, which can be broken down into G1, S, and G2, and cell division, which is made up of mitosis and cytokinesis. A cell spends most of its life in interphase. In G1, it grows and develops. S is DNA synthesis, when DNA is replicated. And G2 does the final preparations for cell division. In cell division, mitosis is the division of the nucleus, and cytokinesis is the division of the rest of the cell, including the cytoplasm. All of this put together creates a cell cycle, which is directed by internal controls or checkpoints in the G1, G2, and mitosis stages. Each checkpoint is of particular significance because it checks for certain crucial events to have occurred before the go signal is given for cell division. Two regulatory proteins in the cycle are kinase and cisplatin. Kinase is always abundant, but is only activated when cisplatin bonds to it. Because of this, kinases are called cisplatin-dependent kinase, or CDKs. A CDK and a cisplatin-bonded protein is called an MPF complex, which is mitosis promoting factor, because it triggers the start of cell division. MPFs are important in determining a cell's passage through a checkpoint. Also, in the pro-metaphase stage of mitosis, MPFs help fragment the nucleus and create spindle fibers. At checkpoints, signals evaluate whether crucial cellular processes have occurred. G1 is the hardest to get through. If it does not pass, it may enter the G0 phase, in which the cell never divides. Some human cells, like muscle cells and nerve cells, are always in this phase. G2 this checkpoint is passed when enough MPFs are produced to initiate cell division. In the M phase, cell division does not continue unless all the chromosomes are properly attached to spindle fibers. These are all internal signals. External signals includes the lack of a certain nutrient or the absence of a growth factor, which is a protein released by cells stimulating other cells to divide. An example of this would be platelet-derived growth factor, PDGF, which is made by blood cell fragments called platelets. PDGF signals cells by binding to a cell surface receptor, tyrosine kinase. This allows cells to pass the G1 checkpoint. Another external factor is density-dependent inhibition, a phenomenon in which crowded cells stop dividing. Also, anchorage dependence, which is the requirement that animal cells need to divide, they must be attached to a substratum, such as the extracellular matrix of a tissue. Cancer cells have lost these cycle control mechanisms and do not respond to normal regulatory symbols. Cancer cells have disrupted cell cycle control systems and indefinitely divide, making them immortal. Cancer cells also have the ability to transform normal cells into infected ones.